Dear colleagues, first of all, I would like to thank organizers for providing me with this opportunity to present our data at this nice conference. I hope you enjoy it. You know that one of the most interesting discoveries done at the intersection of different disciplines, across different disciplines. And one of them is dendrogenomics, which integrates dendrochronology, dendroecology, dendroclimatology, and genomics data. So, and this term dendrogenomics was coined in one of our earlier papers. You also know that trees respond to the environment and to climatic events such as drought, pest outbreak, extreme freezing, not the cold seasons, cold periods, and it's reflected in their triennial ring growth, which can be measured by extracting and scanning wood cores, which could be also cross-dated and assigned to particular events, such as droughts, for instance. So, and this data can be associated with genomic data. So, the important dendrophenotypes can be inferred from this dendrochronological data, such as resistance, recovery, resilience, and Ivan talked about resilience, gave a nice presentation. So here's an example for 5 drought periods in large populations. So, and through cross-dating, you can assign some tree rings to particular periods, and measuring tree rings before drought and during drought and after drought, and making different ratios, you can estimate several parameters or indexes such as resistance, recovery, resilience. If you measure, for instance, tree ring 3 years before, take average growth during 3 previous years before drought, and if you measure during drought and after, take a ratio measuring during drought to previous, that would represent resistance, and so forth. Some other important dendrophenotypes can be also measured, like average tree ring width or basal area increment, which could be a proxy for biomass and heterosis. Heterosis measured as a growth rate. Also, variance of tree ring width and basal area increment can be also measured, and that could be a proxy for homeostasis, for stability of individual development. Like you assume that with less variance, it means that trees are less dependent on the environment and have more stable individual development. So all these individual dendrophenotypes can be a variation of these phenotypes can be associated with genetic variation. So, most presentation of this conference so far were devoted to the temperate forest, but you know that boreal forests represent about 25–35% of the entire world forest. So, and 80% of this forest in Eurasia and about maybe 70% in Siberia. So mostly it's represented by conifers. It's mostly conifers, conifer forests. And here in this slide, I present 4 the most common like keystone conifers in boreal forests, and Siberian stone pine, Scots pine, Norway spruce, and Siberian larch. So, and here were sized and arranged not according to their height, but according to their genome size in comparison with human genome. So, and you can see that they have huge genomes like Siberian stone pine, or also European stone pine, Zambra pine has genome almost 10 times larger than human genome. So, and here for this particular conifers, we have genome assemblies or draft assemblies, which are publicly available for Norway spruce, Siberian larch, partially available for Scots pine. So, and but in this presentation, I will focus, I have data also for Siberian stone pine and Scots pine, but here I will focus, because of limited time, I will focus on Siberian larch, which has the largest, or one of the largest area in Eurasia, or Asia, which actually is the second only to Scots pine. It's a kind of typical conifer with relatively small cones and wind-propagated small seeds. But unlike all other conifers in the family Pinaceae, it's the only one which has seasonal senescence, which is an interesting feature. We also analyzed genetic mechanisms of this. We sequenced, assembled, and fully annotated Siberian larch genome, and we benefit from this. So analyzing different genetic mechanisms. Also, chloroplast genome is available, which was easy. It's easy now to sequence and assemble chloroplast genome. They are relatively small. But mitogenome, mitochondrial genomes is a different story because it's not circular. It consists of multiple forms, multiple subcircles, linear forms, not necessarily circles. So some of them could be concatenated. It's actually even more difficult to assemble mitochondrial genome. We succeeded for Siberian larch, published it recently, and it's now the largest. And if you compare the human genome, which is very small, only 16,000 base pairs, 16Ks, this is 11 million base pairs. But even if you have incomplete sequence, a genome sequence, it allows to identify and develop a lot of microsatellite, species-specific microsatellite loci. We did it for Siberian pine, for larch, for Scots pine, and we intentionally focused on developing microsatellite markers with long repeats, which could be easily genotyped using even simple gel electrophoresis. So, which very handy if you do field research, field study, and you need to genotype really fast some polymorphic markers in the field conditions. So you don't need capillary electrophoresis. But we developed sets for both for simple genotyping and for capillary electrophoresis. And one of our early studies was using genetic data based on microsatellite markers and dendrochronology data. We looked for associations between complex individual parameters, genetic parameters such as heterozygosity, to see how it's associated with some dendrophenotypes, because that could be used as a proxy for homeostasis. And heterosis, assuming that high heterozygosity could be associated with more stable development and with heterosis. We didn't find these associations in that particular study where we analyzed two Siberian larch populations in Altai-Sayan mountain region in Siberia, East Siberia. But we found some interesting we observed some interesting phenomena that fast-growing trees had high annual growth variance. They're kind of more climate sensitive, they're more sensitive to environmental variables, such as precipitation, temperature, and others. When we also used dendrochronology data and genomic markers, now we used to study mountain hemlock, to study tree line of mountain hemlock in Alaska, to see migration, to infer recruitment, because it's also a very important question, how that would ecotone would behave in marginal population, because what we see due to climate warming, that tree line moving toward north. And it's interesting to see how it's sustainable. Is it supported by recruitment, by long distance, by local recruitment, or by long distance migration? So we, using dendrochronology data, we aged all trees. That was pretty exhaustive sampling on with one of the areas, Kenai Peninsula in Alaska. So, and we used now more genome-wide markers, which were genotyped using genotyping by sequencing or restriction enzyme-associated DNA polymorphism approach. So we found some interesting observations and see how it's recruited that can help to make some management, conservation management decision. So you are familiar with this approach, genotyping by sequencing. We used double digestion restriction enzyme DNA associated sequencing. We tested several restriction enzymes, because at that time we didn't know, we didn't have reference genome, because now you can, through in silico computer modeling, to select the most optimal combination of restriction enzymes for red sequencing. So, but at that time we didn't know, so we tested several. We choose the best pair for the most optimal to amplify about 1% of the entire genome using this genome reduction approach. That would allow us to identify about 170,000 SNPs, but after filtering and quality control and some presence in most trees, we selected about 5,000 SNPs, which was actually sufficient to infer kinship and relatedness between individual trees and use it in our analysis. So more recently, we analyzed 5 populations of Siberian larch using the same genotyping approach, double digestion red sequencing. In 5 populations, 130 trees per population. And finally, 136 of them were used for analysis. We inferred dendrophenotypes, such as resistance, recovery, resilience, and others, and associated them with about 11,000 high-quality SNPs using 5 drought periods. And here in this slide, you can see our workflow, which actually present our pipeline, which was set at our genomic cluster at the supercomputer center. So we, after isolation of DNA sequencing, after double digestion treatment and multiplexing, ligation adapters, sequencing, when sequences were processed for quality, for demultiplexing, reads were aligned to the reference genome of Siberian larch. On the right side of this slide, you can see publications which we use the data from these studies we used in this analysis, like reference genome for nuclear genome, for mitochondrial genome chloroplast, to partition our sequences into different type of genomes. Also, we found all transcription start sites in promoter regions, because it's very important, because SNPs which are associated with transcription start sites can significantly affect gene expression and can much more affect phenotypes than even some non-synonymous substitution in exons, in coding regions. So through SNP calling, when we used also population structure analysis and search for associations using different programs, Bayesian space linear mix model and some other programs, we also looked for association with environmental variables, but I'm not going to talk about this here. I have very limited time. So this pipeline is installed and working nicely for our data, and we are open to collaboration with any other who would be interested in analysis like this. So briefly about 11,000 SNPs were discovered, genotyped, and using genome-wide association study, we found some associations with some loci where they were annotated. And we also actually used the outlier approach to find some outliers, those markers which variation and differentiation cannot be explained by selectively neutral markers. So you also know, all of you know this approach. But for association study, we analyzed to take into account confounding factors. We analyzed population structure. We found two or three major clusters, where you can also see in this PCA chart how populations are distributed in cluster analysis. So that was taken into account in mixed model analysis as a confounding factor. We also used kinship into account, kinship relationships. So, and finally, we identified a few interesting candidate genes whose variation is likely to be adaptive, because strongly associated with dendrophenotype variation. We also analyzed gene ontology enrichment for particular functions or molecular classes for particular genes. So we found, no time to go into detail, but I would be happy to provide them. So finally, there's a conclusion. Conclusions are pretty straightforward, that individual and population response to climatic factors and different biotic and abiotic stresses can be and was registered in Siberian larch and Siberian pine trees using our dendrogenomics approach. And it depends on location, because these climatic responses are much more pronounced in the population growing in the extreme environmental conditions. It was a little bit problematic for Siberian stone pine to find climatic response because, okay, that's a different story. Anyway, this GWAS analysis allowed us to identify several important candidate genes with functions which could be related to these adaptive traits. And that actually allows us to find adaptive potential, which is important in conservation programs, or for predictions for assisted migrations also would be important to take into account standing adaptive potential of trees. So we found many SNPs which were associated, but actually many of them were associated with promoter regions and even with transcription start sites, which is very interesting, we think. But we found not too many associations. We expected more, and we think that it could be due to sample size. Certainly, the sample size should be larger, and more SNPs should be analyzed. So those who are interested in this approach, a recent review was published, relatively recent. You can read this, with where our data and other data published, publicly available data are presented and summarized. and discussed. So I would like to acknowledge our dendrogenomics team from from Kharkaz Technical Institute and the Siberian Federal University. I would like to acknowledge Laboratory for Genomics and Department of Genomics and Bioinformatics of Siberian Federal University. So, and separately, I would like to acknowledge our computing and bioinformatics team. You know that it's very important to have a good bioinformaticians with a good computers, and we benefited from using Supercomputer Center with a nice computer scientists and bioinformaticians. And separately, I would like to acknowledge our DNA sequencing team. So I would like to acknowledge for funding, and thank you for your attention. I would be glad to take your questions.
Thank you very much, Kostya. Are there any questions? Yes, Christian.
Hi, Kostya.
Hi.
Sorry if I missed it. Did you use natural populations or common gardens for your dendrophenotypes? And if it's natural populations, how did you correct for, or how did you account for the influence of the different environments in the natural populations in the GWAS?
Thank you. It's a good question. We use natural populations in this presentation. Only natural populations, Siberian large populations in Sayan and Altai mountain regions. We also have data, of course, for environmental variables, for bioclimatic, like nights in typical, bioclimatic variables. We also analyzed association of dendrophenotypes with variables, so we see contribution of them. We didn't use them as confounding factor because we shouldn't, because we're looking for adaptive variation. We use selectively neutral variation to control for false positive, false negative associations. But of course, we analyzed the contribution of environmental variables into variation which we observed. The short answer, yes. So we use bioclimatic variables, but it's for different presentation. So because that's a lot of data also. And some interesting, we analyzed, we especially interested in association with altitude, because Siberian larch is interesting species. That's a northmost species, which is the only conifer which grows close to, even behind polar circle. So it's well adapted to freezing, to very harsh environmental conditions, but it also grows at different altitude, as well as Siberian stone pine. So we analyzed in association with altitude, we found interesting genetic mechanisms of adaptation to grow in harsh condition at high altitude. Did I answer your question?
Yes, but I just wanted to note that I think, in my opinion, you have to account somehow for environment in GWAS in wild populations, because you could, it's not only the genotype that influences your phenotype, but also the environment. And I think it's very challenging to do that, and we need to find somehow a solution how to do that.
If you use it as confounding factor, you can kill your real association with adaptive variation, because they could be co-correlated.
I think this is the case when you use climate in a common garden as a covariant. Then you remove the influence of the environment on the phenotype. But in wild populations, you have this difference in the environment, and you should somehow incorporate it. But I'm happy to talk with you about that. Not here.
Yeah, it's a good point. Yes, we have to discuss it. And yeah, you might be right. Thank you.
Hi, thank you for your talk. I was curious if you could go into a little bit about the methodology of how you identify neutral SNPs.
Okay. Yes, actually, the slide which sorry, it's a different slide. Yes, we, through mapping, because we have full annotation, very detailed annotation of Siberian larch, including promoter region and transcription start sites. We selected SNPs which are not associated, which are not in contigs or scaffolds with any genes. So representing intergenic regions, and supposedly variation is selectively neutral since we are not associated through linkage disequilibrium with any other functional genes. Of course, we cannot exclude that some of them still could be associated with some important genetic elements, but we selected 700, sorry, that was wrong. Can you see my screen? I'll go back to this. Yes, here we use 761 supposedly selectively neutral SNPs. These SNPs represent those SNPs which are located in non-coding regions, at least based on our annotation. I think most of them should be selectively neutral. Some of them, yes. So that's how we use this population structure as confounding factor to avoid false associations, to control for false association. Did I answer your question? Thank you.
Thanks for the very interesting talk. My question has to do with the sample size. You mentioned that it's very important, especially in order to show small effect SNPs. Can you tell us a bit more about this? How many trees and how this sampling was organized, or how you sampled?
Well, it's this kind of preliminary study, because like a proof of concept, because certainly you would need much larger. All simulations shows when you have quantitative traits with multiple gene control, if a small contribution of each, you need a very large population size. You also need more markers to find some associations. So certainly the SNPs which we found, we just may be lucky. And because what is 11,000 SNPs for 12 gigabase genome size? It's 1 per million, roughly, 1 SNP per million. So you miss a lot of potentially important associations. So, and also what I see, that's interesting, what I see in most studies where genome-wise is based even on full genome sequencing, most SNPs contribute a little bit. But if you look at SNPs which are associated with promoter regions or transcription start sites, the story is different, because they significantly affect expression, gene expression, and that can contribute much more. Not like a few percent for each SNP which we observed in most genome-wide association studies, but you can see 10, 15, and even more percent. So that's, we have to focus and pay more attention to SNPs which are located in promoter regions, which can affect transcription factors and transcription start sites.